Go ahead, Bazad.
Hello, everyone, and welcome to today's seminar. It is my pleasure to introduce today's speaker, Dr. Remnao Tian. He's an accomplished researcher in microbiology. He obtained his PhD from the Hong Kong University of Science and Technology in 2015, where he studied the diversity and function of microbes in different environments. From 2016 to 2019, as a postdoc research associate at the University of Oklahoma, he worked on several projects funded by the Department of Energy with a particular focus on microbiomes in soil, water, and animal guts. In 2019, Remnao joined iFish at the Illinois Tech as a research scientist. He has been instrumental in the development of several bioinformatic tools and using machine learning techniques. His extensive experience in both experimental and computational biology has enabled him conduct and lead research on microbial diversity, function, and ecology using cutting-edge tools and techniques in genomics, metagenomics, and metatranscriptomics. He's a skillful programmer, good at machine learning techniques, and very experienced in the lab, especially with the high throughput sequencing technology in different platforms, for example, Illumina and Oxford Nanopore sequencers. In short, he is a great scientist and researcher in the field of environmental microbiology, with over 70 publications in high-impact scientific journals. Remnao, please go ahead.
Okay, thank you for the introduction from Bazad. Hello, everyone. Ladies and gentlemen, welcome to the seminar. Before we get started, I would like to ask a soul-searching question. Do you worry that AI will take your job? Well, honestly, I am a little worried. I hope my presentation will help you get a good answer and get you relieved. Okay. Okay, let's get started. And the topic of today's seminar is application of artificial intelligence, which is AI, and machine learning, ML, in microbiological research.
Remnao, could you share your screen? We don't see your PowerPoint.
Oh, sorry.
No problem.
So let's share.
Do you still have it up from our dry run earlier?
Yeah, I closed it and restarted. Okay, yeah.
Hi, Rue.
Okay, did you see it?
Great, yeah.
Good to go. Put it in your slideshow, hon.
Yeah.
Great.
Right.
Thank you. Okay, sorry, I didn't show this slides. Yeah, and there's a soul-searching question. Do you worry that AI will take your job? And I hope my presentation will help you get a good answer. Okay, let's get started. And the title, application of AI and ML in microbiological research. I am Remnao Tian. I'm a research scientist from iFish. Okay, first I would like to give you some introduction of AI and ML. What is AI? This is a very common word these days. And AI stands for artificial intelligence, which refers to the ability of machines to simulate human intelligence. Note that it's simulating human intelligence. And ML stands for machine learning, which is a subset of AI. And it enables machines to learn from data without being explicitly programmed. Here, what is explicitly programmed? We can see here, there's a robot learning some stuff. And the explicitly programmed stuff, it means you tell the machine how to calculate something. Like, for example, if you like to get a volume of a cube, you give it an equation, so it do the math. So this is a explicitly programmed. But machine learning is not like this. Machine learning is like you feed it with a training data, but you don't tell it how to calculate those variables. It just do it by itself. So this is the some concept of AI, ML, and explicitly programmed. And although AI and ML has a very brief history, it can be dated back to like mid 20th century, almost like 17 years old, the same age as a computer, right? But it is actually very important in today's world. So I'm gonna show you a video to show how.
Somehow there is no voice, I think.
The voice.
You may need to exit the presentation mode to get it to play right.
Okay, maybe we can skip it, and generally it shows the sorting and packaging of those stuff when you are shopping online. And do you know that before that when you are doing online shopping, your stuff was sent by AI actually. Okay, and next, we see that it is actually very important in today's world. We see it everywhere, even when you are doing online shopping. So how it becomes so important to us? So we can see the impact of AI and ML here. First, we look at the benefits of AI and ML. It has AI and ML have the potential to bring many benefits to the society, such as increased efficiency, improved decision making, and better quality of life. But however, they also raise some risks and challenges, such as job displacement. Look at here, the robot is asking the human to go away, and also such as the bias and discrimination and privacy and security concerns. Moreover, there are ethical considerations that must be taken into account, such as fairness, transparency, and accountability. Therefore, it is important to develop and deploy AI and ML in a responsible way and ethical manner to ensure that their benefits are shared widely. here I put a picture of the robot Sophia here to show the ethical issue. This is the robot Sophia. What if she wants to get married or get divorced? There are some ethical issues, right? So we see that there are many impacts of AI. So how does it impact us? We can look at the application of AI and ML. So where can it be applied? Here are some examples, like transportation. And in transportation, AI and ML can be used for autonomous vehicles. Like here, this is a Tesla self-driving car. It can recognize the objects on the road and do the corresponding response using this model, machine learning model. And it also can, AI can also be used for traffic prediction and logistic optimization. And in healthcare, AI and ML can be used for disease diagnosis, drug discovery, and personalized treatment. Here is an example of the surgery robots. They can do the surgery by themselves sometimes. In finance, AI and ML can be used for fraud detection, risk assessment, and investment management. And like in manufacturing, AI can be used for predictive maintenance, quality control, and supply chain management, and so on. And like entertainment, education, and even research. So you guys must know that what's this, right? This is the ChatGPT, which becomes very popular in recent months. And I use ChatGPT for do the coding work, and it's very useful to write some scripts and even develop an app. So it's useful, but I would like to ask, has it been used in research? You know, we are research guys, and I do a little investigation. I search machine learning in the NCBI's PubMed to look for those publications with the doing machine learning actually. And here's an example of that, a guide to machine learning for biologists. And as a result, there are more than 100,000 publications since 1957 year. We can see the very beginning paper, 1957. But during right after that, there's actually a very small number of papers until the recent 10 years. We can see here, it is exponentially growing, and it is doubling every 2.5 years. And in the year 2022, there are more than 30,000 publications related to machine learning. So it has become very popular in the recent 3 years. So it is actually a new field to apply ML in biology and microbiology. So guess what? We are in the golden time. So here I would like, in this topic, I would like to talk about the machine learning application in microbiological research. First, I would like to introduce what is microbiology. Microbiology is a science to study microorganisms, including like bacteria, archaea, virus, and protozoa. And why is important? Why is it important? Microbiology. It is important to understand microorganisms' roles in human health, disease, and like agriculture and the environment. So what does machine learning? What's the role of machine learning in biological research? Okay, it can accelerate the research progress. It can improve the data analysis progress. And also it can enable a more precise prediction in microbiology. We'll see it later. And so now machine learning has been started to be applied in microbiological research. Here are some examples. Like it can do species classification. I put a picture here to show how to classify the species using machine learning. And basically, it needs you need to feed it with training data. Like here, you can feed it using the morphological data, like the phenotype, its shape, or color, things like that. And the machine then use the machine learning to model how to classify a sample based on these features, and then it can give you the prediction. One of the predictions is the classification. So it is very good at classifying species, actually. And it can also be used for functional gene classification, such as you can predict DNA sequence if it what's the gene name, what's the function it has, and also it can predict antibiotic resistance. Like you can feed it with some features of the bacteria, like some genes, functional profile, and then it can predict whether this bacteria will resist an antibiotic like penicillin. And also it can predict the microbial phenotype, and it can help developing drugs, and it can help predict pathogen, which is one of our projects currently. And today, I'm gonna show you an example of our project which use machine learning to predict molecule types. By molecule types, I mean whether this molecule is a chromosome or plasmid. Okay, here is the title of our project, Plasmid Hunter: Accurate and Fast Prediction on Plasmid Sequences using Gene Content Profile and Machine Learning. And this is the logo of our software. And yeah, first some introduction to plasmids. I don't know if you people know plasmids. And plasmids are a small DNA molecule which is separated from the chromosome of a bacteria or archaea. You see here is the main part of the genome, the chromosome, and these are the plasmids, which is separated from the chromosome, and they are usually circular and double-stranded DNA that can autonomously replicate within the host cell. Actually, they are very common in bacteria and archaea and even eukaryote. And why do bacteria need plasmids? It is because plasmid can carry non-essential and beneficial genes that can help their host to tolerate some harsh environment and help them survive. And also, plasmid play a key role in spreading microbial resistance amongst bacteria species, and which pose a threat to animal and human health. So why do we need plasmids? Yeah, plasmids. Some plasmid-borne genes can be used to explore, to be utilized in genetic engineering. Like we can use plasmid to clone a gene and transform it to another host cell and to express it. It is a very important genetic engineering tool. And moreover, plasmids harbors many gene clusters for secondary metabolites that can be used for the development of like drugs or natural products, including new antibiotics. So plasmid is actually very important to both bacteria and human in research. So but there are some challenges in plasmid studies. Like currently we have some available tools to study plasmids, such as the high throughput sequencing HTS, which means using some platform like Illumina to sequence DNA. They have very high throughput, so each time we can get a huge amount of DNA sequences, and they can be used for whole genome sequencing, WGS. And what is whole genome sequencing? That is to sequence a genome using a strategy like shotgun. We can cut or shear the genome DNA into small pieces as a library, then sequence each small fragment, and then use them to assemble them into the original genome sequence. This is called WGS, whole genome sequencing. And by the assembly and finishing, we can get a whole genome and whole plasmid. Then we can determine which DNA sequence is a chromosome or plasmid based on the DNA sequencing size. Like, for example, if you have like 3 mega base pair, then we know that it is a chromosome, because usually a plasmid is smaller than like 1 megabase pair, usually like 200 kb. This is one of the important strategies, but most of the time we don't finish a genome because it's very time consuming and costly. Most of the time we only do the WGS and assemble it to a draft genome. What is a draft genome? A draft genome means it is only assembled but not finished. You only have many long fragments, and they are called contigs. So you can never tell a contig is a chromosome or plasmid based on the size, because they are not complete. They are just a fragment of the chromosome or plasmid. So this is the challenge in draft genome and even metagenome. You cannot get the complete chromosome and plasmids. So to solve this problem, we develop machine learning tools to predict those DNA sequences, whether they are plasmid or chromosome. Okay, here's our workflow. I'm gonna briefly describe these processes. First, we download the database from NCBI's RefSeq database, which is a very big database. It is a curated one, curated by NCBI, and the counterpart of the RefSeq is the GenBank database, which is not curated. And we downloaded this database. It has 25,000 complete genomes, and we know that which is chromosome and which is plasmid. Then we use the protein sequence of those genomes, in total 36 million proteins, and we process these by doing clustering and removing the singletons, and then we get almost 1 million representative sequences, and we use it as a database for gene annotation. And on the other hand, we collect those chromosome and plasmid sequences, and to make a training data, we simulate contigs from them, which are some fragments of different lengths, and then we use the database mentioned before to annotate them and get the gene content profiles, and then we make a training data set. A training data set is a feature table. It has many samples, and for each samples you have the features of each gene, the absence and presence, and then you know which this sample is a chromosome or plasmid. This is the training data set, and then we do the machine learning modeling and and also do the validation and get the best model. And after that, we get another set of the chromosome plasmids as the test data. We also simulate contigs from the test data and also get the gene profile as the feature, and then use the best model to predict them, and then do the evaluation. So to evaluate this accuracy in many using many indicators, and also we compare our data, our tools, our models with other tools, and do the benchmarking. Okay, we can see the results here. First, about the performance of our model, Plasmid Hunter. We actually used a couple of algorithms of machine learning, different algorithms, and including the random forest, decision tree, the naive Bayes, the logistic regression, and other, and also we include another batch using the PCA transformed data, using the same algorithm. So among all these different algorithms, we found that the NB, naive Bayes, has the best performance. In these indicators, we can see one by one. The first indicator is the accuracy. Accuracy means how accurately your model can be in predicting the test samples. For example, if you have 100 test samples and you predict correctly for 90 samples, then you have an accuracy of 90%. So in terms of accuracy, we can see the green bars here, which is the naive Bayes, has the highest accuracy, which is almost 96%, which is actually a very high accuracy in the field of machine learning. And this is the accuracy. And the second indicator is the second evaluation is the balanced accuracy. Balanced accuracy means if your test data is not balanced, for example, if you have 100 samples for test, but there are like 10 plasmids and 90 chromosomes, then that is a imbalanced data. So that is not fair for the model to predict. So you need to balance it. So the second indicator is balanced accuracy, because we use the balanced test data. So the balanced accuracy is the same as the raw accuracy. Okay, the third indicator is the log loss. The log loss evaluates how confident your model is. For example, if your model predict one sample correctly with a very high confidence, like it is 99% confident that it is a plasmid, and then this log loss will be very high. If you predict the sample correctly, but you are not that confident, you are only like 60% confident, then this log loss will be lower. So this indicator evaluates your confidence, how confident you are, and of course, the higher the better. And we can see that our naive Bayes is not bad. It's better than the other two here. It is very sorry. The log loss is the lower the better. I mean, the lower, that means you are more confident. You are less likely to make a mistake. So we are the green bar is actually low enough. Okay, the fourth indicator is the recall. Recall is you must have heard a term sensitivity, and how sensitive is your model to predict the positive samples. So the recall is the same thing. The recall means it is calculated by the 2 positive over the 2 positive plus the false negative. It means if you have a 100 positive samples, and if your model predict 90 of them, then the recall or sensitivity is 90%. This evaluation indicates how sensitive your model is. And precision means how much of your positive prediction is correct. For example, if you predict 100 samples as positive, and 90 of them are correct, then it is 90% precision. So in both here recall and precision, the green bars are quite high. In recall, it is also like more than 95%. That is more than 95% sensitive. And the precision is also not bad. It's also, yes, more than 95%. And the next indicator, F score, it is a harmonic mean of the recall and precision. Basically, it is a balanced indicator of this of the previous 2 indicators, and as a result, the green bar is the highest one here. And the last indicator is the ROC AUC, and this ROC is the it evaluates the the reliability of a model when you are using different cutoff, prediction cutoff. And here is the ROC curve. If the x-axis is the false positive and the y-axis is the true positive, it shows when you're using different prediction cutoff, what is the true positive and false positive. So, and the AUC means the area under the curve. And usually the area, the larger, the better. So we can see that the green lines here has very good AUC, the area. You can see here the green bar is the second highest one. So considering all these indicators comprehensively, we choose the NB, the naive Bayes, as our final model to do the prediction. And this is a comparison to other tools. There are currently some other machine learning models to do the same job, to predict plasmid from draft genome or metagenome sequences, and like PlasClass, PlasForest, deep plasmids, and ours is this one, Plasmid Hunter, the blue one. If we can see in terms of accuracy, it is in different. We also in the benchmarking, we use different contexts with different lengths, including 5 kb, 10 kb, 20 kb, 50, and 100 kb, because this modeling is very impacted by the sequencing length, very to a great extent. It's easy to understand. You have a DNA sequence that is very short, like 1 kb. You have very limited information about this, right? So it's hard to predict based on a very short DNA. But otherwise, if you have a very long one, like 100 kb, you have enough information, and it's easier for you to predict it. So we need to challenge the model with the short sequences, like we use the 5, 10, 20, 50, and 100 kb. So as a result, we can see here in terms of the accuracy, the blue ones is our tools. It beat all the other ones. It is the highest one in all the lengths. And the highest one is the 100 kb. It is more than 96% accurate. And the other ones, except these two, are a little lower, but these two are much lower, especially for the longer sequences. And in terms of recall, ours is also the best one. It's similar to the accuracy here. And in terms of precision, ours for the shorter sequences, the blue line is lower than the other three, but in turn for the longer sequences, our tool is still very good, almost the same as the highest one. And to get a balanced evaluation of the recall and precision, we look at the F-score here, which shows our tool is the best for all the contigu lengths. And we also evaluate how fast the models can run. As a result, we can see our tools can finish those data set within 8 minutes for all the lengths. And some of the other tools are quite fast, like in 2 minutes, 3 minutes. but some others are very slow, like 10 minutes and even 100 minutes. So ours is not bad in the middle, but considering the accuracy and other indicator, ours is outperforms the others. So we also discussed why does Plasmid Hunters stand out from the other tools. We explained it in 2 points. The first point is how we select the features as the training data. We select the gene content profile, and we consider it has a higher resolution than the other indicator. What does the other tools use for the feature? Some of them use the Kramer frequency. Kramer frequencies means the relative abundance of continuous nucleotide like ATCG-AT or TGCA, and it is the relative abundance of these sequences in your samples, and this Kramer profile is actually have lower resolution. It only has, for example, the K4 only has 200 possible recombination. But for the gene content profile, it is very variable. We have like more than 10,000 different genes, right? So this feature has a higher resolution, and we actually tested the effects of the gene content profile and the Kramer profile. You can see here using the Kramer 4, 5, 6 to cluster the simulated context. We can see that the chromosome, which is the blue circles, and the plasmids are the orange circles. They are mixed together. It's hard to distinguish that for all the Kramer profile. But for gene content, we can see here, they are very well separated. The blue ones are the chromosome, and the plasmid orange. You can see here we zoom this area as this here. We can see that all the chromosomes are clustered together, and the plasmid are far away from that. So this shows the resolution of the gene content profile is higher than Kramer. That's why our tools can predict very accurately. And another point is that it is that we use simple features. Some of the other tools use very many kinds of features, like including Kramer, and also include other features like GC content and sequences length and some other indicators. They are using too much features. Actually, there is a law in machine learning. It's called Occam's razor law, and it is also called the law of parsimony. It is a principle that states that among competing explanations that predict the same results, the simplest explanation is more likely to be correct. In other words, when there are multiple possible explanations of a phenomenon, the one that requires the fewest assumptions or entities is usually the best explanation. And Occam's razor is often used in scientific inquiry and problem solving, or where simpler theories or models are favored over more complex ones. So all other things being equal. When so so we can see that the simple features are better. We use the simple. We only use one indicator or feature. That is gene content profile. So okay, so we have for this project we have finished it and written the manuscript and has submitted to the journal Genome Biology. And we also submitted to the bioRxiv, which is a preprint platform, and you can access to it on bioRxiv by searching Plasmid Hunter. And we also released the CoLab platform on Google. You can, if you want to use it, you can get the link from this this manuscript, and you can run the CoLab scripts to predict your sequences. And okay, I think that's all of my presentation, and I would like to acknowledge Bezar Emmanuel, who is my supervisor. Thank you for his support and supervision. And I would like to thank IFISH and FDA for their support. And lastly, I thank all machine learning developers and engineers, which make my project possible. And so let's get back to the very beginning, the question. Do you really worry that AI will take your jobs? I am a little worried because I am doing the data analysis and like programming. So we can see whose jobs will be taken by AI. Yeah, there are some jobs that involving repetitive tasks or data analysis may be at high risk for automation by AI. However, jobs that require creativity or critical thinking or emotional intelligence and interpersonal skills are less likely to be fully replaced by AI. And so you should get relieved if you are doing some creative jobs. And so the last take-home message is that AI is rapidly changing the world, and we cannot stop its progress. And what we do is not fear it, but learn how to take advantage of it. So hurry up, don't wait too long. It's time to learn machine learning. Okay, thank you, everyone.
Thank you very much, Remnao, for the great presentation. If anybody has questions, please just unmute yourself and go ahead, ask Remnao.
Hi, Remnao. This is Idai. Thank you very much. That's really interesting and informative. I have a question back on the workflow slide.
Yeah.
That seems like you separate the sequencing plasmid and chromosome to two subsets, which one is testing set and one's training set.
Right.
Is that correct?
Yes.
So how you determine the portion, like the 3,000 to be the testing side and 15,000 to be the modeling side? Is there a way to determine how much in the subset?
Yeah, the principle is to make sure it is sufficient. For the modeling part, we have 15,000 each. So in total, that's 30,000. We have tested different level from like 1,000 to 2,000, 5,000, 10,000, and we at the high at the 15,000, it has it become more and more accurate and precise. So we take 15,000, which is accurate enough, but not consuming too much of our computational resource.
Okay.
To ensure you have got a good accuracy and also efficiency. And for the testing, 3,000 is a good number to test a model. Usually, like our principle is to have more than 500.
Okay.
And here we use 3,000 to test, and it has a very close accuracy level with the modeling part.
And also, the slides on comparing other two, I realized for the precision result, I think the curves are kind of similar. Is that because the algorithm you use, like the tools used? Not this one, the following one for compare the, the precision.
Yeah.
So
Yeah.
those curves are similar. Is that because the algorithm used?
No, this is the other tools. And except the blue line and the other lines are the other tools. We are using actually different algorithm.
Mm-hmm.
And the similarity do not indicate that they are using the same algorithm. It just means it's the level at different.
Yeah, I was just guessing. Thank you so much.
You're welcome.
Remnao, maybe I can ask you a question.
Yes.
It's a good presentation. Actually, I also not remind me the manuscript that I read before you submit for publication. So I did actually have a couple questions. So one is so this tool that you're using, it's for specific sites of the genome. So especially that you're showing now here, it's between 5 KB to 100 KB, right?
Yeah.
So anything above that 100 KB, it's may not be applicable.
Yeah, we recommend to use this model for context longer than 5 KB because the lower, the accuracy will be lower. We have tested, and it is a common. It is the normal capacity of models. If we have too short, you can get very limited information, so you can get a good prediction. So here we have recommend in the manuscript, we recommend to use 5 KB and longer for the prediction.
Follow up with another question which I asked earlier as well, that relevant to the food safety applications or food safety microbiology. Now, how many of those potential pathogens that we are working on may or may not have a plasmid? I know that many of those like E. coli or maybe like the X bacteria, a lot of them they actually do have plasmids. And also that if if those plasmids, I mean, of those sites are there, then what kind of potential applications in the area that we are working in?
Yeah, in terms of food science, a major part of our project is the pathogen. And for pathogen, usually there are many virulence factors which can be carried by plasmid and or transformed by plasmid from one host to another. So our model can be used to predict plasmids from draft genome or even metagenome to get the plasmid sequences, which usually carrying virulence factors. And after you get the plasmids, you can do further analysis on those virulence factors. This is one of the scenarios for to use our model in food science, food pathogen research. Yeah,
Thank you.
thank you.
Remnao, what is your recommendation for those who are kind of intimidated by machine learning, AI, the complexity of these topics? So where they can start, which is a good start, for lay person.
Okay, yeah, I strongly encourage you guys to start learning machine learning and apply it in your project. And to get started, you can search for some textbook or online stuff to learn the basics. And in terms of the tools, you can start it by using the sklearn package of Python, which is very useful and versatile. And for those advanced users, they usually use deep learning, which use some packages of like PyTorch and some TensorFlow. Those are there are many available tools, especially Python packages. So go to learn machine learning and learn Python, which is very easy to, yeah. Thank you.
Thank you very much. I think we are close to the end of our time for this seminar. Thank you very much, Remnao, and thank you all for joining us. I really appreciate it.
Thank you.
Yes, thank you very much.
Yep, great job, Remnao.
Thank you.
Thank you.
Bye.